In this lesson or in this class, we are going to resume our topic that we were discussing, if you remember, before the lockdown or when the colleges were closed. As you remember, uh, according to our course outline of Genetics 2 of the 8th semester, we have three topics in a row, and the title of these three topics is Mechanism of Genetic Change. Mechanism of Genetic Change 1, Mechanisms of Genetic Change 2, and Mechanisms of Genetic Change 3. Now, we are discussing mechanisms of genetic change 2. We somehow skipped the first part, which was mechanism of genetics 1, gene mutation. In this second part of the mechanisms of genetic change, we were discussing recombination, the general homologous recombinations. We have discussed the holiday model. And now, as you can see, the slide in front of you, it's site-specific recombination, or what they call the conservative site-specific recombination. As you can see from its title or the name of this particular recombination, that this is specific recombination which takes place at a very specific site within the DNA molecule. Therefore, it is also known as conservative. Now, by specific, we usually mean that whenever a recombination will take place in DNA, it will take place at that particular point, at that particular sequence of the base pairs, and not anywhere else. So this is why it is known as site specific. It is not general, what we have discussed before. However, since it's a recombination, so the usual process of recombination is that the DNA strands break and then exchange parts and then they rejoin. And similar to general recombination, there must be a certain degree of sequence homology between the two sequences that are being exchanged. Now, it is commonly also known as SSRS, site-specific. The enzymes for it are known as site-specific recombinases. Site-specific recombinases. So, as the name suggests, it is not one enzyme. There are many enzymes, so they are known as site-specific recombinases. What these enzymes do, they bind to those sites which are short DNA sequences. These site-specific recombinase enzymes, they recognize those sequences, they bind to it, and then they cleavage the DNA molecule. After the cleavage, since it's a recombination, so exchange of DNA segments or small portions of the DNA molecules are exchanged, and when the exchange is complete, the final part is the rejoining of the DNA strands, so the DNA molecules become intact. But now they are in a recombined state, or what we say that recombination has taken place. And since it takes place at a very specific point, at a very specific part or location of the DNA molecule and not anywhere else, so this is known as conservative or site-specific recombination. Site-specific recombinations require, as we have just seen, various recombinase enzymes. However, some of the site-specific recombinations require molecules other than site-specific recombinases. So they are usually known as accessory proteins. Also, we have some multiple genome modification strategies, and these are the recombinase mediated cassette exchange RMCE. Now we are going to discuss it shortly, that what these cassettes are, what do they mean. The site-specific recombinations, they are employed or used or they occur in various cellular processes like bacterial genome replication, differentiation, pathogenesis, and movement of mobile genetic elements. So these site-specific recombinations, uh, they can also be helpful for genetic engineers, or they can be used as genetic engineering tools. Now, what may be the size of the recombination sites, whether they are long stretches of DNA molecules, long portions of DNA molecules, or small ones. So as you can see, they are quite small, or the portions of the DNA where recombination takes place, they are between 30 to 200 nucleotides. And they contain two special motifs or portions or parts which have an inverted repeat symmetry. So in one strand, the sequence will be say ATGA or something like that, and on the other we have the same sequence in inverted, and these are the sites to which the recombinase binds, and then flank a central crossover sequence at which the recombination takes place. The pairs of sites between which recombination occurs, mostly they are identical, but this is not the case always. Like ATTP and ATTB of lambda integrase the lambda phage. Now this is a diagram of now one of the best examples of site-specific recombination are the lambda phages. You all know, and you must have studied lysogeny. Lysogenic cycle of the virus. It is usually the lambda phage who carries out this lysogenic cycle. Now this lambda phage, when its DNA enters the host cell or the host bacterial cell, it gets itself incorporated, or it enters the host DNA at a very specific site. So it can initiate, or because of this prophage or the DNA of this lambda phage, recombination might take place, or what we call the site specific. Now this particular diagram we have taken from Molecular Biology of Genes, 7th edition, chapter 12, which is dedicated to this site specific recombinations. Now this particular diagram is showing how or what is the outcome of recombination. Now, as you can see, there are three possible outcomes of site-specific recombination. One of the mechanism, which is the first one on the left, a DNA molecule is inserted. In the second one, as you can see, the DNA molecule is removed. So the first one is insertion. In the second recombination, we get a new molecule which is smaller than the parent molecule or the original molecule, because a part of the DNA is deleted, it is removed. And in the third case of site-specific recombination, the DNA which is inserted or DNA which is present already, it may get inverted. So an inversion event occurs. So in site-specific recombination, we might get a new DNA molecule inserted into it, or the DNA molecule may be, a part of the DNA molecule may be removed, so we get a deletion, or the DNA molecule may be inverted.